Hello, my name is Mikayla Spinler, and I'm a current PhD student at Kansas State University studying applied swine nutrition. Today, I'll be talking about my study looking at split suckling strategies on lifetime growth performance and mortality. This study was a part of the Improving Pigs Livability Grant. When we think about split suckling is commonly used in large litters, and the goal of split suckling is to ensure that all piglets within a litter have an opportunity to consume adequate levels of colostrum, with the overall goal of split suckling being to decrease pre-wean mortality by giving those pigs enough colostrum. However, when we compare split suckling protocols from different farms, these protocols vary greatly, as well as the farm's ability to successfully implement these split suckling protocols. And really, when we look at the published literature on split suckling, it doesn't indicate one best strategy that leads to the best results of overall decreasing pre mortality. And the published literature shows a wide range in how these split suckling protocols are implemented. There's been 7 trials published from 1996 to 2023 looking at different split suckling strategies. And again, when we look across these 7 different trials, they do not indicate one best protocol that leads to the best results of decreasing pre-mortality. So when we look at these 7 different studies, 2 of these studies found that split suckling decreased pre-mortality, while 5 of these studies actually found no differences in pre-mortality. Of the studies that reported pre-weaning growth performance, 1 study found that split suckling actually decreased pre-wean growth performance, while 3 found no differences, and 1 found that it tended to improve pre-weaning growth performance. So really, when we look across these 7 different trials, they vary in the number of pigs that are pulled off during split suckling, how long these pigs are pulled off during split suckling, as well as how many times, if they just do one split suckling, or if they pull these pigs off 2 or 3 more times. And it also varies in how they select which pigs are split suckled, with the majority of studies selecting pigs for split suckling based on either birth order or body weight. Because when we look at the overall literature on split suckling, it does not indicate one best strategy. We really wanted to compare 2 different split suckling strategies and see how they affected both pre and post-weaning litter performance. So for this study, we conducted the study at a commercial farm in Nebraska and used 1,500 mixed parity sows with an average parity of 3.6 and their litters with roughly 22,800 piglets in total. Sows and their litters were allotted to one of 3 treatments, and we'll get into more detail on our split suckle treatments in the following slide and give you a visual of how these treatments were implemented. But our first treatment was our control, and these litters were not split suckled during this trial. Our second treatment was split suckling based on, and for this treatment, the first 8 pigs born in the litter were marked as they were born. Once the sow was done farrowing, we removed the first 8 born pigs for 45 minutes. And then after that 45 minutes, we did a swap where we put the first 8 born pigs back on the sow and then removed the later born pigs in that litter for 45 minutes. Our third treatment was split suckling based on body weight. And for this treatment, after the sow was done farrowing, we removed the 8 heaviest pigs from the litter for 1.5 hours. For this study, it's important to note that we only used litters that had 10 or more piglets in this study, and we cross-fostered within treatment after the split suckling treatment was applied and after birth weights were taken within 24 hours after the birth of the first pig in that litter. So to dig into our 2 different split suckle treatments a little bit more, we'll first start with split suckling based on birth order. So for this treatment, as the sow was actively farrowing, we marked the first 8 pigs born. And after that sow was done farrowing, and we counted her as being done farrowing when afterbirth was passed, we put up our split suckle board here that ran from one side of the crate to the other. And we put our first 8 born pigs, removed them from the sow, and put them on the other side of our split suckle board under a heat lamp in the crate. And removed these first 8 born pigs for 45 minutes. After that 45-minute time frame was up, we put the first 8 born pigs back on the sow. And then we pulled off the later born pigs in that litter, the unmarked later-born pigs. We removed these later-born pigs for 45 minutes, and then after that 45-minute time frame was up, we put all piglets back on the sow. And that was the completion of our split suckle treatment based on birth order. For our second split suckle treatment, we split suckled based on body weight. And for this treatment, after the sow was done farrowing, we removed the 8 heaviest pigs from that sow and placed them under a heat lamp in the crate. We removed these 8 heaviest pigs for 1.5 hours. And after that time frame was up, the 8 heaviest pigs were placed back on the sow, and that was the completion of this split suckle treatment. For this treatment, we did not do any swapping, and we only removed one set of pigs, the 8 heaviest pigs, for 1.5 hours. For this study, we took individual pig weights prior to split suckling and at the time of weaning. On a subset of litters, roughly 45 per treatment, we took blood samples on whole litters and a weight 24 hours after the birth of the first pig in that litter. And we took these blood samples and looked at immunocute ratio to give us an indication of colostrum intake. We recorded the pig ID, date, and reason for any mortality, and we also identified and removed fallback pigs within a litter and recorded the date and weight when these fallback pigs were removed from the litter and placed onto nurse sows. Fallback pigs were removed from the litter anytime between day 2 post-farrowing and day 12. On a subset of pigs, we fall them into the nursery and the finisher to track lifetime growth performance and mortality to really see if our split suckle treatment had an impact on downstream growth performance or mortality of these pigs. Now we're going to dig into the data from this study, and I'll take a moment to set up our slides for you. So as a reminder of our treatment, in the purple bars will be our control treatment, and these litters were not split suckled. In our green balls will be split suckling based on birth order. And for this, the first 8 born pigs were removed from the sow for 45 minutes. After that 45 minutes was up, the first 8 born pigs were placed back on the sow, and the later born pigs were removed for 45 minutes. Our third treatment was split suckling based on body weight. And for this treatment, we removed the 8 heaviest pigs from the sow for 1.5 hours. After that 1.5 hour time frame was up, the 8 heaviest pigs were placed back on the sow. As I mentioned earlier, really when we think about split suckling, one of the overall goals of split suckling is to ensure that all piglets in a litter consume adequate colostrum, hoping that this leads to reductions in pre-wean mortality. So for this study, we broke our pre-wean mortality into different time frames, day 1 to 2, and then day 2 to weaning. So this day 1 to 2 and day 2 to weaning would only represent piglets that were weighed and tagged at the time that birth weights were taken, and would not include any pigs that died prior to this time point. This pre-wean mortality would also not include any pigs that were removed and placed onto nurse sows. So for this study, when we look at pre-wean mortality from day 1 to 2, we do not see any differences across our 3 different treatments. We also did not see any differences in pre-wean mortality from day 2 to weaning. So overall, when we look at the overall population in our study, split suckling did not affect pre-wean mortality of our pigs. We really wanted to get in this data a little bit farther, and because we took individual weights of our pigs, we were able to look at pre-wean mortality based on body weight at day 1 of the study. We broke our pigs into roughly 3 equal body weight categories. We have first our light body weight category, being any pig born less than 2.6 pounds. Then we have our medium body weight category, those pigs weighing 2.7 to 3.2 pounds at birth. And then we have our heavy body weight category, those pigs being 3.3 pounds or greater at day 1. And when we look across our 3 different body weight categories, we again see no differences in pre-wean mortality among our 3 different treatments. As I mentioned earlier, fallback pigs were identified and they were removed and placed onto nurse sows from day 2 post-farrowing until day 12. And when we look across our 3 different treatments, we again see no differences in the percentage of pigs that were removed and placed onto nurse sows among our 3 different treatments. When we look at litter size on day 1, or the time of weighing and tagging, and then again at wean, we again see no differences among our 3 different treatments in litter size. We took birth weights of these pigs at day 1 or the start of the study, and then again at weaning. And when we look across our 3 different treatments, we again see no differences due to split suckling treatment on pig body weight at weaning. We took blood samples on whole litters, 45 per treatment, to look at immunocrit ratio. And immunocrit ratio can give us an indication of colostrum intake. So we really use this immunocrit ratio to try and understand if our split suckling treatment was having an impact on colostrum intake of our pigs. Immunocrit ratio measures the level of immunoglobulins in the serum of these pigs. And immunoglobulins are produced by the immune system and help to protect pigs from bacteria and different viruses. And piglets, when they're first born, they receive these immunoglobulins from colostrum. So because our immunocrit ratio measures the immunoglobulin level in the serum, we can use that as an indication of colostrum intake, with a higher immunocrit ratio indicating a higher colostrum intake. However, when we look at immunocrit ratio across our 3 different treatments, we do not see any differences, indicating that likely our split suckle treatment did not have an impact on colostrum intake of our pigs. We also took a 24-hour weight of these pigs when we took that blood sample. So when we took our blood sample, we did not see any differences in gain from day 1 to 2, or the time from weighing and the initial weighing and tagging until 24 hours post weighing and tagging. We did not see any differences in piglet gain from day 1 to 2 across our 3 different treatments. An important part of this study, and something that hasn't been reported in many of the papers published on split suckling, is looking at the downstream growth performance of these pigs. We really wanted to understand if split suckling had an impact on our pigs in either the nursery or the finisher. So for this study, we followed 2,200 weaned pigs into the nursery, and these pigs were split equally across our 3 different treatments. In our nursery, we were able to track feed intake on a pen level, and we were able to record average daily gain and feed efficiency by taking a weight of these pigs at the end of the nursery period. For our finisher, we followed a subset of our pigs that we fall into the nursery and into the finisher. And it's important to note that we selected the pigs that we fall into both the nursery and the finisher because these were the pigs that we took those intensive collections of blood samples and day 2 weights on. So we followed these same pigs into both the nursery and then a subset of those pigs into the finisher. With 882 pigs being followed into the finisher, split equally across our 3 different treatments. We've penned our pigs by treatment in both the nursery and the finisher. And again, in the finisher, we were able to measure feed intake on a individual pig level, and we're able to calculate average daily gain and feed efficiency of these pigs by taking a weight before the first marking event in the finisher. And when we look at both our nursery and our finisher data, we see no differences in growth performance or mortality of these pigs, indicating that our split suckle treatment did not have an impact on performance in either the nursery or the finisher. However, we really wanted to dig into this data a little bit farther to really understand, does split suckling have an impact on a subset of our population? Can we find certain litters, piglets, or sows that by applying our split suckling treatment, we have an impact on growth performance or decrease in breed mortality? So we broke our sows into different parity categories: our parity 1, parity 2, parity 3 and 4, and our parity 5 plus sows. And we looked at the interaction between treatment and parity to try and see if we have an impact on a certain parity by applying a split suckle treatment. And we saw no differences and no interaction between treatment and parity. We also looked at the timing of split suckling the same day that litter was born versus split suckling the day after that litter was born. And again, we see no differences in or no interactions between our treatment and split suckle time, indicating that the timing of split suckling did not have an impact. We also looked at functional teat count of our sows. So we broke our sows into 2 categories: sows that had 14 or less versus 15 or more functional teats. And we again see no interactions between treatment and functional teat count. We did break our sows down and we looked at only sows that had a greater pig count at day 1 compared to functional teat count, or these sows had less teats and they had pigs in their litter at day 1. So we pulled out this subset of the population to try and understand if split suckling had an impact on those large litters where we have more pigs than we have functional teats. And when we look at pre-mortality, we do see a difference from day 1 to 2, with our control sows having a higher pre-mortality compared to those that were split suckled based on birth order, with those split suckled based on body weight being intermediate between the two. And when we look at the main reason that these control litters had a higher pre-mortality from day 1 to 2, the main reason is that they had a higher number of pigs that were laid on from day 1 to 2. However, when we look at overall pre-mortality from day 1 to weaning, we do not see any significant differences among our 3 different treatments. It's important to note that this subset of the population will represent roughly half of the sows included in this study. So overall for this study, the split suckling strategies that we investigated and how we applied these split suckling treatments did not result in any differences in pre or post weaning growth performance or mortality. So this really begs the question, if we're not having an impact by split suckling, are there better ways that we can invest time in our sow farms to improve piglet livability? I would like to thank DNA Genetics and Pillen Family Farm for their cooperation and use of their pigs and facility in this study. I would also like to thank all of the amazing interns and farm staff that I got to work with during this study. I would not have been able to do it without them. I'd also like to thank National Pork Board and the Foundation for Food and Agriculture Research for their support of this study, as well as the Improving Pig Livability Grant. If you have more questions about this study or other studies included in the Pig Livability Project, please visit piglivability.org. Thank you for your time today.